Hi, everyone. I'm Saniya Khullar. Currently, I'm a postdoc at the Wellcome Sanger Institute and University of Cambridge. I'm grateful to share one of my PhD projects called NetREm with you today. The paper on NetREm is titled NetREm, Network Regression Embeddings Reveal Cell Type Transcription Factor Coordination for Gene Regulation. It's published in the Bioinformatics Advances journal. So single nucleotide polymorphisms, SNPs, they're the most common type of genetic variation. So SNP RS123 is associated with the DNA base change to C at this spot. Over 90% of disease risk SNPs are non-coding and often affect transcription of genes. So understanding gene regulation can help link genotype to phenotype. Transcription factors are proteins that help activate or repress the expression of target genes, TGs. TFs have DNA sequence-specific motifs that recognize and bind to TF binding sites, TFBSs, along the promoter or enhancer of their TGs, just like two puzzle pieces. Here, a SNP may alter expression of this TG by preventing this TF from binding to its TFBS. So gene regulatory networks, GRNs, they show the directed relationships between many TFs and many TGs. GRNs vary across cell and tissue types, and they change during disease. In eukaryotes, gene regulation is complex, pun intended. You can see direct and indirect protein-protein interactions among many proteins like TFs to form a module to regulate TG transcription. Most TFs, they don't act alone. They work in concert, often cooperatively, occasionally antagonistically. This is the backbone of TF-TF coordination. It's a widespread phenomenon, but it is poorly understood at the cell type level. On the right is a human PPI network from string of direct and indirect functional associations among 8 core TFs in human Schwann cells. Sadly, current PPI networks are global. They are not cell type specific. Plus, they typically use positive weights for all PPIs, even antagonistic ones. Tools like TF cluster, they construct cell type interactomes, but they struggle to explain how gene regulation occurs. Other tools like CNIC+, they infer cell type GRNs, but they treat TFs in isolation. They ignore the interdependent TF-TF coordination networks. RTN duals, it tries to uncover TF co-regulatory behavior, but it does not incorporate prior knowledge. Also, it may sometimes fail to yield results. So we can use data, expression, motif information, chromatin interactions, DNA accessibility to link potential TFs to regulatory elements and TGs. So this cell type GRN will tell us which TFs might regulate our TG. But how do TFs work together to co-regulate the TG? None of this multi-omics data explicitly models TF-TF interactions. So a promising solution is to incorporate prior biological knowledge of TF-TF PPIs. Well, how do we integrate PPI networks with existing input data to uncover cell type gene regulatory mechanisms, both GRNs and TF-TF coordination networks? So how do we do that? So to address this gap, we developed NetREm to uncover how TFs coordinate to co-regulate TGs. It is run step by step for each of the GTGs in a context. I'll walk you through it. So for each TG, NetREm integrates multimodal data. The first input provides the signal, so gene expression data from M samples. TG expression is our response variable Y, and expression levels of N candidate TFs form our predictor matrix X. The second input provides the structure. It's a positive weighted TF-TF PPI network that defines the relationships among predictors. So NetREm can be run just using these two inputs with a fixed list of TFs across all TGs. But we strongly recommend including a prior cell type GRN. While optional, this input acts as a feature selection step, refining the TF list to those most biologically relevant for the TG. So we can use many public PPI databases. Some are dummy, others are weighted based on interaction confidence. And what's exciting is that as PPI networks improve, thanks to research by gurus in the field and all of you, NetREM's performance will only improve, which is great. So traditional GRN methods, they predict TG expression Y from individual TF levels, and they ignore these vital TF-TF interactions. Instead, NetREM treats TFs as collaborators, not as lone wolves. Specifically, NetREM uses network regularized regression. It integrates prior knowledge of PPIs among TFs as a network constraint to guide its regression. So it finds optimal TF coefficients, C star, to predict Y based on both expression and PPI connectivity for TFs. So this allows NetREM to effectively model not only which TFs co-regulate TG, but also how TFs work together to coordinate TG regulation. And if available, we can use other omics data to construct a prior GRN and then input this to NetREM to enhance its biological interpretability and its relevance. So network regularized regression has been proven effective across many biological domains. Even imperfect prior networks can guide learning and enhance the relevance of results. But what sets NetREM apart is its use of regression embeddings, which are latent, hidden representations that combine expression data and prior network structure among predictors. So NetREM uses network regularized regression and embedding-based learning to uncover cell type TF-TF coordination that drives TG expression. Pipeline defines the network regularized regression problem with 3 terms: a data fitting term, a sparsity term, so higher alpha is fewer final TFs, and a network regularization term. So TFs strongly connected in the PPI network tend to have similar coefficient magnitudes. It's a smoothing to guide grouped variable selection among connected TFs. So higher beta leads to stronger network influence. And then step 2, NetREM uses singular value decomposition, produces gene embeddings, X tilde, Y tilde, feeds them C star. And for TG, NetREM outputs 2 key networks. The first is a directed TF to TG regulatory network with positive weights for positive coefficients for activators, negative for repressors. And the second is an undirected TG specific TF-TF coordination network, B, that captures how TFs cooperate, positive, or antagonize, negative, in PPI subnetworks to co-regulate TG. And we can stitch together these outputs across all the TGs to form a comprehensive cell type GRN and to yield a cell type TF-TF coordination network, B bar, assigned percentile from -100 to 100. So basically for each cell type, we have these 3 outputs: a cell type gene regulatory network, and then 2 networks related to TF-TF coordination. One is TG specific. For each of the genes, we have the TF coordination values, and then we have an aggregate value that is cell type TF-TF coordination. And there are more details, and I'm happy to stick around if you have questions. But NetREM is an open source tool on GitHub with detailed documentation, and I'll be putting up tutorials on my YouTube channel, so please stay posted. So we benchmarked NetREM's GRN and TF-TF coordination networks in 10 real-world applications in humans, mice, and yeast. So I highlight some of the results, and the rest are in the paper. You can see the various applications. So here, this is an example. So we evaluate 5 NetREM models where we vary the beta network hyperparameter against several state-of-the-art GRN tools. Here in yeast cell, you can see that no GRN method dominates, but NetREM models perform competitively with top scores, so low ranks, on several metrics, So what sets NetREM apart is that unlike other tools, it also captures TF-TF coordination, both overall and per TG. So in hematopoietic stem cells, HSCs, we run NetREM. This is another example for each TG using 178 candidate TFs, and we compare predictions against a gold standard GRN. So I show 2 representative examples. So here, for these 2 TGs, ElasticNet and Lasso, they return no TFs, and linear regression enriched select all, which shows alarming overfitting. But NetREM makes precise predictions. So for ATF2, 7 of 8 TFs predicted by NetREm are validated. And I also want to show our grouped variable selection property. It prioritizes TF to TF PPI subnetworks that are biologically meaningful. And this sets a stage for future discoveries. So here for RNF167, one of the novel TFs NetREm discovers is TFAP4. And TFAP4, it strongly interacts with validated TFs, as you see here, in key HSC pathways like RB1, which is cell cycle control, T-cell differentiation, and proliferation. So these are key pathways that are really important for hematopoietic stem cells. So this kind of shows how NetREm can prioritize these core TFs. So to summarize our benchmarking of GRNs, NetREm achieves higher precision in signed GRN inference. It's comparable to top GRN tools. It has a grouped variable selection property, and it's robust to changes in the input PPI network. So we benchmark top-ranked TF-TF links from our coordination networks across many data sets. And we find that our top links are enriched in a contextual PPI database. So this suggests that NetREm may help capture cell type PPIs. These are largely unknown, and this can help an unmet need. And one-sided t-tests show that NetREm recovers true PPIs and highlights promising novel links for follow-ups. Next, we evaluate the top K links from 13 NetREm models, so each with a different input PPI network, and from RT and Dual's tool. And we find that 10 NetREm models predict significantly more valid links found in a gold standard evaluation PPI network. So this shows that NetREm outperforms RT and Dual's tool. So we apply NetREm to predict cell type GRN and coordination networks in Schwann cells and Alzheimer's disease versus controls. In both cases, we input a learned prior reference GRN to NetREm. And we identify regulatory modules distinguishing myelinating from non-myelinating Schwann cells. And for AD, which is a neurodegenerative disorder, we use TF-TF coordination networks in ML models to identify cell type neurodegeneration biomarker, TGs. So Schwann cells, they are the myelin sheath along axons of neurons in the peripheral nervous system. So as you see here, these are Schwann cells. And understanding GRNs and TF coordination and myelinating and non-myelinating Schwann cells is actually an open challenge. And it's vital for finding potential therapeutic targets for demyelinating diseases and neuropathies. So NetREm helps uncover these following insights, gene regulatory links and coordination links. And for Schwann cell EQTLs, expression quantitative trait loci, we use tibial nerve data as a proxy. And I'll be focusing on 8 core TFs with strong external validation data found in mice and rats. And their direct target genes are supported based on ChIP-seq evidence of TF binding and loss of function data where knocking out the TF significantly alters TG expression. So both of these figures are truncated. The originals are in the paper, but I wanted to just show that on the left, each point is a direct TG of a TF, and the x-axis shows their expression correlation. And correlations between TFs and their direct target genes are often low. It's comparable to random gene pairs, especially in noisy single-cell data. Yet NetREm can still recover true regulatory relations. For example, SOX10 shows a weak correlation with its direct target gene, CYPA1L2, but NetREm ranks SOX10 as its top 17th TF. And on the right, we analyze direct, loss of function, and novel TGs for these TFs. And the top row uses EQTLs to validate predictions across these TFs. And the bottom fully breaks down the predicted TGs. So in non-myelinating Schwann cells, hypergeometric tests, reveal significant enrichment marked with a star for loss of function TGs for direct TGs for 5 out of 7 TFs. And so this shows NetREm's ability to uncover meaningful TF to TG relationships. And here's an example where I validate a novel SOX10 target gene, FADI1, using epigenomic tracks from rat sciatic nerve. They have SOX10 ChIP-seq peaks and active enhancer marks. and human validation. And then I also do gene enrichment analysis for EGR2 TGs and SOX10 target genes. And they are relevant to Schwann cell identity and function. And this shows NetREm can uncover novel regulatory programs for core TFs. And then here I highlight novel TF-TF coordination links and how those predicted by NetREm are validated experimentally. So in our comprehensive input network, RxRG does not have any known PPIs with YY1, TEAD1, and STAT1. However, NetREm predicts meaningful coordination scores for RxRG with all 3 TFs. For example, RxRG YY1 ranks as the 24th percentile in non-myelinating Schwann cells, which is meaningful despite no prior PPI evidence. And so then we dig deeper and we find that all 4 TFs, they co-regulate 174 TGs and non-myelinating Schwann cells. This network is simplified just to those with validation. And then, for instance, we can zoom in on CDH19 using cut and run tracks and show that these are enhancer regions where all 3 TFs co-localize, and they have EQTL support for this as well. And then RxRG, YY1, it's not a support, like these are not known PPIs, but we have evidence supporting NetREM's predictions of their coordination. So on the left, cut and run heat maps show read density overlaps. So the first is YY1 read centered on SOX10 peaks, which is a known PPI. The second shows this novel link, and which is validated. And on the right, peak overlap analysis across Schwann cell TFs shows that 44% of RxRG peaks overlap with YY1 binding. It's the highest overlap. TEAD1 and YY1 show 24% and 28% overlap with RxRG respectively. So this just supports that NetREm is able to uncover novel TF-TF interactions through coordinated binding, which is key. And then I want to also just quickly show that we can apply NetREm to AD versus controls across 8 cell types. And NetREM's embeddings can capture how TFs coordinate for each TG in a given cell type. And this data can help nominate potential biomarker genes for diseases. So I use a contextual PPI database. I treat the problem as positive unlabeled learning. No neurodegenerative TF links are positives. And then I undersample the negatives because those could be unknown. So both classes are balanced. For each cell type, I calculate how coordination scores differ between AD and controls for each TG. Then I train random forest models to predict which links are associated, TF links with neurodegeneration. All 8 models perform well with CV AUCs of 0.81 or higher. And then we perform enrichment analysis. And then this is just a quick example that just shows that we can go from genotype to phenotype with NetREM. The paper explains this further. So to summarize, NetREM helps address key limitations in state-of-the-art tools. It builds cell type coordination networks, it creates cell type GRNs, and you just need gene expression data and prior knowledge on PPIs. And there are further directions we can go with this. But I just wanted to say thank you to everybody.
Questions? Okay, maybe I can start. I have a question regarding the fact that you are using a prior network, GRN. What if you have in your system very important interactions that are not in the prior? Do you think you can also use that to try to find those new interactions?
Yeah, so for the prior, what I recommend is usually the way I do it is I use motif info, peak to gene links. You just, because you can have like a bunch of cell type TFs, like you could have like 200, 300. So like the goal would be maybe to truncate it down as much. It's just feature selection. You can truncate it down to like 70, 8, it can vary for each TG. But your goal is that, if you have like 1600 TFs, you might want to truncate it already using cell type TFs to 200. But then if you can just like truncate it even more to like what you think are biologically plausible, it could help the model, because there could be TFs that just don't have that, that just might be meaningless, for that. So it's just like, it's up to you if you're like, which TFs you definitely don't think would ever have any way of regulating it.
Okay, thank you.
Hi.
Yes.
I wonder if you have problems, when you put that regularization term related to the prior, you're really encouraging kind of all transcription factor neighborhoods to all participate. So do you have a problem that you tend to kind of bias towards suggesting that there's too many transcription factors involved, especially for a transcription factor that has many interactions with other transcription factors? How do you deal? You see that, and how do you deal with that?
Oh, that's what that's a very good question. So that is what the regression problem inherently the model definition incorporates. That some TFs may be strongly connected. It's like we have a normalization, which is based on like the degree, like how many other PPIs each given TF could be connected towards. So it's kind of like normalizing for the fact that some TFs could be hubs and could be strongly connected to many TFs. So it's already inherently like kind of accounting for that in the regression problem.
Do you see bias? Like your prior should bias you in some way to suggest that the transcription factors involved are not. Do you see that, or do you?
I think for the most part, what it's doing is it is prioritizing like which TFs could regulate the TG based on expression, but it's also trying to prioritize like individual TFs that might not have strong expression correlations, but could be strongly linked to TFs in the network. So it's basically trying to prioritize TF groupings in the network. Yes, it does prioritize like strongly connected TFs. It'll tend to prioritize those, but it's a parameter that you can tune accordingly. Like you can set beta to any like non-negative non-zero number, and then you can tune it to see the results. But I do find that we're able to capture meaningful PPI links that are found in, that are actually known. So I think it is helpful.
So thanks, we have to move
Okay.
on. This
Thank
could
you.
be a discussion for lunch.
Oh yeah, thank you.